In this small village near the Swiss Alps, farmers raise livestock on grassy green pastures. One sunny day in early summer, a Japanese man is strolling through this meadow.
I think it was around here.
He seems to be looking for something.
Ah, here it is. This is a new species of plant known as Insueta.
This plant, of the genus Cardamom, is Cardamom insueta. It's a new plant species that evolved in the 20th century. This person is not some hiker who got lost in the meadow while looking for flowers. He's Professor Kentaro Shimizu, an evolutionary biologist at the University of Zurich. Not only is he conducting field surveys, he's also decoding minute genetic information in the plant's cells. This allows him to examine the mechanisms of plant evolution and diversity. Why does Shimizu pursue these sorts of topics? That's simply because of his inquisitive nature.
Most of the organisms in the world have the same number of genes. That's only about 20,000 to 30,000 at most. Despite that fact, there are so many different species of organisms living on the Earth. Through my research, I want to understand why this happens.
Indeed, his research may help save the future of our Earth. Shimizu is currently working with researchers from around the world to decode and analyze the genes of wheat, one of the world's 3 major grains. The goal is to create a new variety of wheat that can cope with global warming. The project's research is making rapid progress.
We are working on our analysis so that others can use the genetic information as soon as possible. Pretty soon, it will be time to start writing a paper.
In this episode of Science View, we'll follow evolutionary biologist Kentaro Shimizu, who is uncovering the mysteries behind the amazing diversity hidden in plants. Located in the northern part of Switzerland, Zurich lies along a large lake. It's Switzerland's largest city, with a population of about 370,000. The city is home to the University of Zurich, where Albert Einstein received his PhD. This university also hosts the Department of Evolutionary Biology and Environmental Studies, where Shimizu is based. In Shimizu's laboratory, 22 researchers from around the world are trying to unravel the mystery of plant diversity. The university has a full research system in place, with a next-generation sequencer that can read genetic information at high speed, as well as a large-scale computer that can process a vast amount of information. It's in this environment that Shimizu has been studying the new plant species. Shimizu took us to the place where Insueta grows wild. How many times have you gone this year?
This is my first in 2019.
From Zurich, we drive south to an area about an hour and a half away. This is the village of Ernerboden in the canton of Uri. A meadow lies in a valley between craggy mountains. It is here in this small village of just a few dozen people that Insueta was discovered.
We might have a better chance of finding Insueta over there.
Shimizu wasn't the first to discover this plant. Credit for that goes to botanist Elias Landolt, someone Shimizu greatly respects. Landolt conducted surveys in Ernerboden during the 1960s and 70s.
He wrote the book on Swiss botany, so when he came to this village, I think he realized that he'd discovered a new species.
It was in the shape of the leaves that Landolt found a slight difference from plants that were already known. After taking a specimen back to his laboratory, Landolt checked the chromosomes with a microscope. That's when he determined it was a new species. Landolt looked at the chromosomes in the cell nucleus. Back then, he could only see the size and number. But now it's possible to decode the DNA in those very chromosomes and read the genetic information contained inside. Inside the DNA, genes contain important information, although it's just a very small amount. By analyzing the genes, Shimizu is examining the mechanism of the plant's evolution. Shimizu wants to find an Insueta plant growing around here.
Ah, there it is. Here's what I was after. This is the new plant, Insueta.
It's a small plant. Its flowers are only about 1 centimeter in diameter and have 4 petals. However, this Insueta has a surprising ability.
What's characteristic about this plant is that it can live both in partly flooded areas and in dry areas. It adapts to places where the environment changes.
So why was it able to develop such an ability? The environmental changes in this village were a significant factor. According to Shimizu, Insueta is the hybrid offspring of a plant that was native to wet habitats and a plant that was native to dry habitats. The village had been covered by forest until the end of the 19th century. It's thought that the two plants crossed and hybridized after humans cleared the forest and turned it into pasture land. In other words, Insueta is a plant that has evolved in just over 100 years. Is that really conceivable?
Many people probably have the impression that evolution takes place over a long period of time. But we've come to realize that, in fact, it's happening over a very short period.
Plants are sensitive to changes in their environment and respond accordingly, which encourages evolution. This is how Insueta developed. After analyzing Insueta, Shimizu found something interesting. Insueta had inherited one set of chromosomes from the plant native to wet habitats and 2 sets of chromosomes from the plant that lived in dry habitats. He found that the plant could use different genes depending on the situation. The water-resistant parent's gene if there was more water in the pasture land, and the parent's gene which is suited to dry areas if the land was dry.
When there are increased genes, this makes it possible to have a division of different functions. Moreover, it also allows for the use of new functions. We're pretty sure that this is what enables evolution into a new organism.
How does heredity occur? And what does it mean to have an increase in genes?
Let me explain by talking about diploid organisms, such as humans. Basically, humans have two sets of chromosomal DNA, with one set each coming from the father and the mother. This is what defines a diploid organism. And in the process of making a child, the chromosomes are reduced, meaning that the egg cell and sperm will only have one set of chromosomes. Supposing that the egg cell is here on the left and the sperm is on the right, they will fuse and have both sets of chromosomes. This is the process of fertilization. The new organism has two sets of chromosomes. So this is a diploid.
So an organism with two sets of chromosomes is a diploid.
Yes, that's right.
Most diploid organisms, including humans, go on repeating this process of becoming diploids. However, when different species cross, it doesn't work out so well.
As for what happens when crossing with another species, when the egg and sperm cells are formed, each are reduced to one set of chromosomes. Then, when the egg is fertilized, there is one black set and one red set.
However, this new organism cannot produce egg cells or sperm, so the next generation will not be born and the organism cannot evolve.
However, in the natural world, there's a mechanism whereby these different species can combine, and that's known as polyploidization.
This happens when the chromosomes in the egg and sperm are not reduced, so they each retain their two sets. It actually occurs to a certain extent. After fertilization, the new organism ends up with 4 sets of chromosomes. This is known as a tetraploid. When species combine and chromosomes increase, this is called polyploidization. Insueta inherits 1 set of chromosomes from one parent and 2 sets of chromosomes from the other parent. This makes Insueta a triploid. In fact, recent genetic research has revealed that many organisms have experienced polyploidization in the past.
It was a pretty surprising result. According to results obtained by many other research groups, including our own, we've learned that the Earth experienced polyploidization about 65 million years ago. The catalyst for this was the period of mass extinction, when there was a sharp decline in biodiversity. The environment was probably extremely severe, with dinosaurs becoming extinct and most plants dying off toward the end of the Cretaceous period. Since polyploids survived that difficult period, polyploidization is probably what enabled survival in the harsh environment.
Shimizu believes this is indication of how polyploidization is a very important driving force for the evolution of organisms. There was a particular incident that first led Shimizu to think polyploidization was important. That was about 20 years ago. while he was doing fieldwork in Japan's Tottori Prefecture as a grad student at Kyoto University. On Mount Daisen, he happened upon a small plant. A plant that remains important for Shimizu's research even now.
I've been studying a plant here called Arabidopsis kamchatka for nearly 20 years.
This is the plant he encountered in Japan. When he first saw it, Shimizu said he had a strange feeling.
When you keep doing research, about once a year there's a moment when you realize, hey, I found something new. That's really enjoyable. So I found this plant on Mount Daisen, brought it back with me, then tried to put its pollen on the flowers of this type of plant here, called Arabidopsis thaliana, which people have been studying. What happened next was totally unexpected. A hybrid developed, and we cultivated the next generation plant. This really took me by surprise.
From that point on, Shimizu became devoted to studying Arabidopsis kamchatka. He discovered that the plant is a tetraploid and that its parental species grow in cold and warm areas. That gives Arabidopsis kamchatka an incredibly wide range of habitat. Why is that? Shimizu had a flash of inspiration. What if the key to plant diversity is the number of chromosomes? Shimizu has many researchers supporting his efforts. Here, Shimizu is discussing work with Masaomi Hatakeyama. He's an essential member for the decoding of genetic information. Even with the latest machines, only bits and pieces of the enormous amount of information contained in DNA can be read. All of these pieces have to be connected correctly. That's where an information science expert like Hatakeyama comes in. He showed us one of the latest instruments for reading DNA, the next generation sequencer.
This here is the latest sequencer. There are many holes inside this. We put the DNA into them and then the sequencer reads it. It can read 2 rows simultaneously, so we can get a large amount of information all at once.
10 years ago, it took about a decade to sequence human DNA. But now, using this equipment, hundreds of people's DNA information can be sequenced in a single day.
With biology today, having information about living things is not enough. Knowing how to process a large amount of information is very important. Going forward, collaboration between biologists and informaticians will be crucial.
On this particular day, the journal club is being held, and all of the staff in the Shimizu lab are participating.
Are we ready? Okay, please start.
The journal club is a meeting where the staff pass around and discuss academic papers from researchers around the world. In addition to the topics brought up, the study session is indispensable for the discussion of various research methods. The Shimizu laboratory has many international members from 7 countries spanning Asia, the Americas, and Europe. The person here reporting his survey results to Shimizu is a Japanese researcher. This is Dr. Yuji Tokumoto, who has just returned from a rainforest in Malaysia. He's studying the mechanism that makes flowers bloom all at once in the rainforest, focusing at the genetic level. What does he think of the Shimizu lab?
First, I'd say it's conducting an incredibly wide range of research. Moreover, researchers in the lab are explaining some really quite complex things in nature at the genetic level, one by one. I think it's a very high-level research lab.
Dr. Hiroyuki Kakui is another researcher from Japan. Kakui's research is also unique. He's searching for the genes involved in pollen production.
I was the first in the world to recently discover a gene that reduces the amount of pollen, but I'm still searching for other genes involved in pollen.
Why did you choose this research lab?
Well, I decided to come here because I was really impressed by Dr. Shimizu's ability as well as the kind of person he is. If I hadn't been able to meet him, I really don't think I would have wanted to come here. He's a very talented researcher.
Stefan Milosavljevic is an Italian-Swiss PhD student.
It's clear to me that Dr. Shimizu is very passionate about his work. When we see his dedication and energy, this in turn helps make us more and more motivated in our own research.
Gwyneth is from the United States. How would you describe Dr. Shimizu?
As a scientist, ideas. Like, he has a lot of ideas. So because our lab is so broad, like, he's really good at kind of being able to kind of pull all of these things together. So yeah, idea maker.
Xu Chongming, who just came here 2 months ago, hails from China.
I feel like he was very busy during this time because it's semester break. He traveled a lot, but he always emailed me like asking how was everything going, and he always remembers. So I feel like very supportive at the very beginning time.
Just like Shimizu's research topics, many diverse and unique researchers have gathered here. This field is used for experiments, including those for an international project in which Shimizu is participating.
Here's where we enter.
Inside, a large amount of wheat is being cultivated.
This here is Norine 61, a representative type of wheat grown in Japan.
This type of wheat was developed in Japan in the mid-20th century. It can grow in both warm and cold regions. The variety is now widely used in Japan.
Here's where we're doing the experiment.
In here, he's testing out how a rise in temperature affects wheat growth. The temperature inside the plastic area is 1 to 2 degrees higher than the outside temperature. He's found that a slight increase in temperature significantly changes the wheat yield. Shimizu says that if global temperatures continue to rise, it may no longer be possible to produce wheat in Japan. Global environmental change has already led to rising temperatures around the world, and dry weather and droughts continue to plague crops. If temperatures rise further, global wheat production could experience a crisis. That's why researchers from different countries have begun collaborating on the 10 Wheat Genomes Project. The project is decoding the current genetic information for wheat in the hopes of creating a new, better breed. This is to avoid a global reduction in the amount of wheat harvested due to environmental change. Shimizu is devoting some of his time to this project. He believes the genetic information of Japan's Norin 61 could be effective. Furthermore, wheat is a polyploid, and Shimizu's research has been a great help. The entire DNA sequence for Norin 61 has been decoded and made available for use around the world.
We also have begun to use this as very important data when we try to analyze just what sort of genes will be effective or ineffective.
Now, Shimizu is working on even more genetic analysis that will help in improving the breed. So, will Shimizu and Japan's Norin 61 be able to save the world?
In the DNA, there's bound to be thousands of things we don't know, so I'd like to decode them going forward.
Shimizu is currently leading a research group at Yokohama City University in Japan. His work already spans the globe. Rather than focusing on just one topic, Shimizu says that he wants to conduct research which covers a broad range of fields. Just like polyploids, he has various talents.